So welcome everyone. Thanks for joining us for this M2D2 talk. Today we have Karin telling us about single cell data and patch effects. So Karin is a PhD student in fragment-based lab at Helmholtz, Munich, and her work focuses on single cell data analysis and representation learning. So yeah, the floor is yours. Thank you.
Well, thank you very much for this nice introduction and also for inviting me to the series. Let me start with motivation on how I started this project. My previous project was aimed at better understanding diabetes. And for that, I combined a large number of diverse mouse pancreatic islet datasets. Approximately 2 years after, I finally had some exciting results, and I was really happy to share them with my collaborators from the endocrinology laboratory. And then they said that the results are interesting, but they also told me that they actually want to treat humans, not mice. And that's how I started comparing my mouse data to human data sets. And as it turns out, integrating mouse and human is much more challenging than just integrating a large number of mouse data sets. Therefore, I started also working on methods for improving integration of substantial batch effects that I'm going to present you today. Now, let's first dive into details of the data that I'm using. My work is focused on looking at individual cells and trying to understand how their heterogeneity contributes to normal tissue function and disease development. So why do we want to look at individual cells rather than tissue in bulk? Where in bulk, we measure all the cells together, which means that we lose some key variability information. For example, if I have a tissue, there will be a lot of different cell types in that tissue. And for example, we might want to know which cell type in particular is associated with the disease. Furthermore, if we were to observe reduced insulin expression within pancreas, we would be interested to know if the insulin is reduced because beta cells that produce the insulin are dying and there is less of beta cells, or if it is because beta cells are producing less insulin overall, but they are not dying, so they're just being dysfunctional. Similarly, it is possible that some of the cells within populations will be heterogeneous. So some of the cells might be producing more or less insulin, or some of the cells might be undergoing cell cycle division. And this is, again, something for which we need measurements on the level of individual cells. Now, if you also go to drug response measurements, which many of you might be interested in, again, as we all know, drug response does not affect all the cells equally, even if that's a homogeneous population. So there might still be cells that by chance respond and some that do not respond. Therefore, again, the measurements in the single cell level are key. So as you can see, having the single cell resolution on hand is key for many important biological questions. Now, let's look a bit more into details how single cell was applied beforehand in drug development, which is one of the most important application domains for single cell. First of all, let's look at the preclinical stage. Here, for example, you can use single cell data to identify cell types that are perturbed, that are dysfunctional in the disease, and also the proteins that might be differentially expressed. Then you might want to choose your model on which you will be studying drugs and disease. And then you might want to ask, do I want to use a mouse model where every individual cell is located within tissue and is surrounded by the vasculature? Or do I want to use organoids, which are coming from human, but are lacking this tissue structure that is, again, affecting a function of individual cells? In the next step, you might have a couple of promising compounds, and you might be interested in which of them is having any side effects. So if you want to target vasculature of the heart, some of the drugs might also target vascular cells in other organs. And for that, again, we need single cell resolution to disentangle vasculatures from other organs from the rest of the tissue in that organ. Of course, a single cell can be also very much useful for clinical stage of drug development. For example, if you look at patient stratification, having a single cell data will provide us much better resolution and precision than the bulk data, therefore providing better stratification. One key example here would be monitoring treatment progression, for example, in cancer, where having only a few cancer cells left will actually potentially lead to de novo onset of the disease. So therefore, having single cell measurements here is really key to detect those individual cells that might be residual after the therapy. So overall, it is very useful to have single cell measurements across the whole spectrum of drug development tasks. Now, we also want to know what kind of information can we actually get about individual cells. And luckily, there's a lot of technologies that enable us to profile a wide spectrum of omic layers, ranging from expression regulation and genetic mutations, that then affect gene expression and protein function, and downstream also metabolism and cell morphology. For that, different technologies were developed, ranging from sequencing to imaging to mass spectrometry. My particular focus is on transcriptomic measurements with sequencing due to multiple reasons. First of all, it allows us to profile a large number of genes, meaning that we get a holistic view of cell function. Second of all, it is relatively high throughput, meaning that we get a lot of cells and therefore can better understand tissue cell population structure. It is also relatively close to the downstream tissue cell function. So that would be, for example, the proteome and the metabolome. And lastly, it is very popular, which means that a lot of data sets have been generated and that there is a great need to develop new technologies that enable analysis of this sequencing modality. Now, because so many data sets were developed, it became clear that actually combining multiple existing data sets can provide us insights that we could not obtain from individual data sets. However, this is also complicated by the batch effects that I'm going to focus in this talk. One example, for example, would be if we have patient stratification, where we might want to pull data sets across different hospitals to get more statistical power. Well, those data sets will be relatively similar in terms of their biological characteristics and also technical properties. There will nevertheless be some batch effects that we need to remove if we want to make those data sets comparable. On the other hand, there are also data sets that have much stronger biological and technical differences. For example, if you wanted to use public data sets for target identification, which is a very promising strategy because there's a lot of public data sets measuring different drugs, different genetic mutations, and different diseases, we are at the same time faced with the challenge that those data sets were generated with different technologies, for example, single cell and single nuclei sequencing, were generated in different model organisms or with different in vitro models such as cell lines or organoids. In that case, we need to compare a lot of different data sets that differ both in biology and technology. Similarly, if we want to select the best possible model system, we will need to compare different models, so animals and organoids, to the primary human tissue. And all these differences are much larger than what you can see on the left. Therefore, I will call them substantial batch effects in the rest of the talk. Now, because of this challenge, and at the same time, great need to enable batch effect removal, a lot of integration methods have been developed, and their popularity has actually increased in the past years. However, those methods don't really perform well when dealing with substantial batch effects, for example, cross-species integration. Therefore, my aim was to improve the integration of substantial batch effects and therefore is cross-system analysis. Now, I'm going to guide you through the process of achieving good data integration. So first, we need to define what is actually a good integration. And I already give you a hint, it relies both on batch correction and biological preservation. Then I'm going to show you some different approaches with which we can improve batch correction and biological preservation. And lastly, I'm going to show you how we combine those approaches to get the best possible integration. Now let's go into more information about how single cell integration actually looks like. Well, what we have at the beginning of every integration is a data set where we have some batch effects, for example, here shown in violet and yellow. And then we have some biological variability, such as cell types that are shown in different shapes. What we want to achieve after integration is to remove batch effects, meaning that the batches are mixed, but still preserve the biological variation, so cell types being separate. Now, how this looks in practice, in single cell analysis, we often use UMAPs to represent single cell transcriptome in 2D so that we can visualize it. And here, every dot would be a cell. What you can see before integration is that the cell types are separating, but so are the batches. And what you see after the integration is the cell types are still nicely separated. However, now the batches are nicely mixed within the cell types. Therefore, the overall goal is to remove batch effects while preserving biological variation. Now, one important question when we do integration is, of course, what are undesired batch effects? And to answer that question, we need to be aware that there are two different axes of variation in our data. One of them is biological axis, for example, donor characteristics such as sex or age, different species, or different in vitro models. The other axis is technical variation, such as sample handling, sequencing protocols, or data preprocessing. The question is, which of those effects do we want to remove? And the answer to that is rather complicated because it really depends on the specific data set and the biological question at hand. For example, if you're interested in comparing different mouse strains to select your model organism, you definitely should not be removing the mouse strain effects from your data because this is what you want to study at the end. However, if you just want to study diabetes, you definitely should remove the effects of different mouse strains so this is not confounding your diabetes research. Therefore, you will need to carefully select what you regard as the batch effects depending on your downstream application. So once we start doing integration, of course, we need to ask, how do we measure if our batch correction is any good? Well, first thing we want to do is remove batch effects, which means that we want to have mixed batches. For that, we can, for example, look at the neighborhood of every cell and check if the neighboring cells are mainly coming from one batch, so orange batch and blue batch, which would be undesired, or if they're actually mixed in the neighborhood, which would be desired, meaning that the batches were nicely mixed and integrated. On the other hand, we also want to preserve true differences because sometimes there might be differences between batches that we don't actually want to remove fully. For example, here we have 3 samples. 2 of them are healthy controls, and for those we would expect that they map on top. And one of them was perturbed with interferon, therefore it is biologically different and we want to map it, that is mapped separately. Now, what we would expect in our UMAP is that the 2 blue batches are overlapping and the red batch is separated. So this type of integration would be good. However, on the top, you can see that all 3 samples are overlapping, which is undesired because we're removing some of the interesting variation. This type of evaluation is, as I already mentioned, kind of very dependent on prior knowledge and specific for a given data set and cell type. Therefore, it is hard to streamline for benchmarking purposes, but is nevertheless important to look at some stage of your integration evaluation. So overall, what we want to achieve in batch correction is to strike the correct balance in batch alignment, not to under-integrate or over-integrate. The second very important question is also that we preserve biological differences because we are doing integration in the first place to then look at the biology in the downstream analysis on the integrated embedding. One thing that is very popular is looking at cell type clusters. So what we have is we have prior cell type annotation, which is here shown in different colors. And then we also have clusters in the data that I kind of encircled here. And you would want to see that this prior cell type annotation matches to the data clusters, which does seem to be the case in the bottom example. However, in the top, actually, you don't see any clear clustering in data. All the cell types are kind of mixed, which means that we are losing this cell type information in the upper clusters. Another important thing to keep in mind is that there might be sometimes more variation in the data than what we have in our prior annotation. For example, here you can see one cell type on which we nevertheless see a gradient of gene expression. And this can be, for example, measured with spatial correlation metrics such as Moran's-I. On the top example, this gradient is lost, so the cells are mixed from all sides of the gradient, which is something that it is undesired. Therefore, to fully understand if our integration is well preserving biological information, we must combine multiple metrics that measure different aspects of biological variation. Now, let's look into how integration can be achieved in practice. There's a lot of methods that have been developed for single-cell integration. I'm going to present you a couple of examples here just to get a feeling of what kind of strategies can be used. But I definitely urge you to look more into the literature if you're generally interested in exploring this field, because this is in no way a comprehensive overview. So one approach is integrative non-negative matrix factorization. What we have here is we have two batches, which are two matrices, each of them being combined of cells that express genes as the features. And then we want to decompose each of those matrices so that part of the composition is specific to that matrix or to the batch, so it represents batch effects, and part of the composition is actually shared across both of the batches. Therefore, we assume that it represents biological effects, and we use this shared representation as our integrated embedding. Another approach relies on mutual nearest neighbors. What happens here is we have two batches, and then we have mutual nearest neighbors between the cells across the batches, and we use this information for aligning of the batches. The last approach that I'm going to focus most on today is based on conditional variational autoencoders. What we do here is we take expression of genes and the batch information, we put it through a neural network, through an encoder, and then when we get to decoder, we again put in the batch information to reconstruct the data beside the latent space, which means because we are now inputting the batch information again, our latent space does not need to contain any batch information and therefore can be regarded as batch-free or our integrated representation. Now, why do I want to focus on cVAEs? Well, because they're in generally very popular choice. They have good performance, they're flexible with respect to the batch structure, so if you have nested batch effects or any other types of metadata that you want to add in different ways to remove, this is very easy to add. And most importantly, for like today's field, because we have so many data sets, they actually scale very well to large data sets and large number of cells. Now, let's look just a bit more into the cVAE architecture, because I'm going to talk about extensions upon that architecture afterwards. So I just want to bring you all on the same page. So what we have for every cell, we have gene expression, which is the input, and the batch information that we put in the encoder. Then for every cell, we would predict the posterior mean and variance. And from that distribution, we would sample latent representation that we would, together with the batch information, put through the coder to predict the gene expression again. To train this network, we have two objectives. One of them is reconstruction objective. It basically checks if the predicted expression is similar to the input expression. And the second one is the regularization objective, which basically checks if our posterior parameters are close to the prior. We need this for two reasons. First of all, to be able to sample the right representation. And second of all, it also helps to regularize the latent space to reduce overfitting and to make internally better behaved latent space, which is very important for single cell analysis, because we're going to use this representation for our cells, and then for base our downstream analysis and a lot of manual work based on this representation, so it is good that it is well behaved. And what we at the end actually use for integrated representation of our cells would be the posterior mean of every cell. Now, just to summarize, what are the key takeaways that you need to remember about single-cell integration for the rest of the talk? First of all, in the integration, we want to remove batch effects and preserve biological variation. And we should be focusing here, or I'll be focusing here on the cVAEs, because they're among the most promising integration methods. Now, let's have a look into what kind of strategy.
Question?
Yeah?
Sorry, just
Yes.
before we move on from integration, can you expand on how you measure, whether this is from the chat, something is under or over integrated?
Yeah, so this is something that you cannot really measure directly because there is no ground knowledge of what is over or unintegrated. So underintegrated would be basically if there is something the batches don't really mix up. And then you could, as I'm going to explain in the rest of the talk also, you could kind of mix the batches to an extreme where all the cells are collapsed together. And that is, of course, also not good because we're removing biological information. So at the end, you basically try to achieve very high batch correction, but also keep your biological preservation high at the same time, and then strike the balance between the two of them. So you never know what is the optimal batch correction, but you try to make sure that both of those metric types are high, and that would be your assumption on being good enough. Yeah, so it's a bit hard to define and also needs some downstream analysis later on, but that's what we usually do, basically striking a balance.
Thanks.
Okay, can I move on, or was that not clear?
That was it.
Okay. Let's then go to different strategies. So the first strategy, that is, I guess, the most popular strategies because it is really part of any cVAE model, is just increasing their regularization objective weight because it is part of normal cVAE. So what happens here? You're basically pushing all your cells towards the prior distribution, which means that your batches will be also mixed together because all the cells are mixed together. On the other hand, what also happens if you do that, also your biological information will be lost because you're really pushing all the cells towards the prior, which is just standard normal Gaussian. And this is what I was also referring to in the answer to the question that, of course, you will get optimal batch removal if everything is mixed together, but this is not what you want to achieve here. So how this looks in practice, and maybe that also helps to explain the question a bit better, is if you increase the regularization objective weight, what will be happening? Your batch correction will increase. However, the biological preservation will also drop. And how this then looks on the UMAP is here we have a low regularization weight. We have nice separation of cell types, but the batches also don't really mix. They're kind of located together, but not really completely mixing. On the other example, here you have higher regularization weight. The cell types are now being pushed together, so we're losing some biological information. However, we also have higher mixing of the batches. So you see the batches are actually mixed within the cell types. Now, one important thing to note about cVAE integration is that it is actually relatively insufficient and low in comparison to other methods that I'm going to explain in the rest of the talk. So what you see here, higher batch correction is better, and I circled the cVAE model across 3 different data sets. You can see it is constantly performing very poorly. And on the right, you can see one selection of integration where I try to select a good balance between batch correction and biological information from the different parameters I tried. And as you can see, the batches are actually not mixed very nicely. Now, because this is such a popular strategy, I wanted to understand a bit better what is actually happening when we are tuning the regularization objective. So as you remember, what we use for our cell representation is the posterior mean. And when we increase regularization, the posterior mean will be pushed towards 0, which means that more and more features will be 0 across all of the cells. So you can also see it here as we increase regularization. Individual dots here are features. Most of the features have relatively low span, and only a few features remain a bit larger. And because it is customary in single cell to use Euclidean distances between cells to then do downstream analysis, basically computing cell neighborhood graph, and on that doing clustering and visualizations and so on, only a few of those features will be actually actively contributing to the distances. Therefore, my question is there any information in the other features that we are missing in that way because we're using the Euclidean distances as is normally done? And as it turns out, if we actually scale those features so that they are all used more equally in learning of distances between cells, it turns out that as we increase regularization, the batch correction is actually not increasing and biological preservation is not really dropping, meaning that the information is still in the feature, so we haven't really removed neither biological nor batch effects, but what we have done is simply drop the number of features that we are using. And you can also see this on the embeddings. So in both cases, both with low and high regularization, the batches are kind of separated. Therefore, I think that the cVAE strategy is not the best one because it literally just discards information regardless of being biological or batch information. And also in general, it does not lead to very high integration that would be needed, for example, when we integrated different species or other systems that have substantial batch effects. Now, to overcome that, one strategy that was previously proposed and actually actively discriminates between batch and biological effects is adversarial learning. And this approach has gained a lot of traction and been added to a lot of single cell integration tools. So what happens here? We have the normal encoder that encodes the cells in the latent space. And then on top of that, we add a discriminator that tries to classify which batch every cell is coming from. So it's driven by a classification loss. And then what we want to achieve is we want to train an encoder in adversarial manner so that it triggers the discriminator that it is not longer able to separate what is the batch identity of every individual cell, which means that the cells will be mixed together and therefore the batch effects will be removed. However, one challenge of this method is that if there are any populations that have unbalanced cell proportions across batches, they will be necessarily mixed together so that the discriminator won't be able to distinguish them. And this is also something that was observed in other distribution alignment approaches where we put together the whole distribution across batches. So in practice, how this looks. So here we have just a normal cVAE integration. I'm showing here just the cell types. And then I added on top of every cell type a number, which is a log ratio between the number of cells between the two batches. So being close to 0 means it's relatively balanced, and being further away from 0 means it is unbalanced. And here I circle the 3 clusters that have the most unbalance. What you can see happens in adversarial learning is those populations will get mixed together. Now, this is something that might not always happen, because on the other hand, we also need to achieve good reconstruction. And if you're mixing together cells that are very biologically distinct, so have very different expression, the reconstruction loss will also drop. However, it happens very often when we look at finer resolution. So within cell type biological variation, because then this mixing will affect the reconstruction loss relatively less, and therefore it's quite common. So what you see here is we have a gene program that is known to be variable in this specific cell type. Here is the non-integrated data showing a nice gradient. Then we have a cVAE. Also, you can see that this gradient is more or less preserved. And then we have our adversarial model. Here I'm using Glue, which is an established model. And you see that these cells are actually mixed together, and also the more enzyme metric that measures spatial covariation is relatively low. Based on those observations, we conclude that adversarial learning is inappropriate for single cell integration because batches very often have unbalanced cell proportions, and therefore it can actually lead to wrong biological interpretations downstream. Now, to overcome this challenge of adversarial learning, we propose to use cycle consistency loss. This is not a new approach, and it has been also used before in single cell community. However, it was always combined together with adversarial learning, therefore its true potential was overlooked. So what happens here? We take a cell, we put it through the encoder, and then when you decode it, we swap the batch information so that we get a counterfactual representation from another batch. Then we input this representation back to the encoder to get a latest representation, and then we push those two together. Therefore, we're always comparing only cells with identical biological background across batches, which means that we won't have mixing of populations with unbalanced proportions across batches as we had in adversarial learning. So to have a quick look in how this looks. So here we have adversarial learning, as if you remember, the cell types were mixed. And here we have a cVAE where those cell types are nicely separated. Another thing, of course, that is important to test if this approach improves upon just a normal cVAE. So I have here 3 different data sets in rows, and then I have different metrics, and then the cycle model is shown in green, and the cVAE is shown in dark pink. As you can see, the biological preservation both on coarse and the fine level is relatively similar between the two models. However, the batch correction is higher when we use the cycle consistency in all 3 data settings. Another thing that you might be able to see here is that the adversarial model glue that is shown in light pink is actually much higher than both of them in batch correction. However, we think that this is overestimated, so it is actually over-integrated because it also loses biological information. This is what I was saying before, how you decide what is the best best integration or what is under over-integration. Well, it also depends on the batch correction, the biological preservation on the other side. So we think that this is probably overestimated and not the optimal batch correction. So to summarize, if we use cycle consistency loss, we are better able to discriminate between biological and batch variation, and therefore we can get more truthful integration. Now to summarize, I would definitely not recommend you to use tuning of the regularization objective with the standard normal prior to remove batch effects, because this will remove also your biological information, and it's also not a very strong method. Adversarial learning might provide you stronger batch integration. However, it will also lead to mixing of cells with unbalanced proportions across batches. And the cycle consistency here is our winner because it has both good batch correction and biological preservation. Now, let's quickly look also through some of the methods that actually improve preservation of biological variation. First of all, we are going to propose to use Venn prior for integration. Now, Venn prior has been before used for single cell representation, and it has been shown that improves biological preservation. However, it hasn't been tested in the integration setting, and I'm going to motivate you why it is so interesting for integration in specifically. But what is actually the Venn prior? So this is a multimodal prior where you have multiple Gaussian components in your prior. So basically something like a Gaussian mixture. However, what separates it from a Gaussian mixture is that the prior parameters are actually learned in the input space. So you have here prior parameters, you pass them through the encoder, and you get their posterior representation. And this posterior representation of the prior of the parameters is actually used as the parameters of the prior. In that way, the prior is directly coupled to the posterior. And what we observe here is, of course, it improves biological variation because we have multimodal priors, so the latent space is more flexible. However, one very unexpected findings we had is that it also improves removal of batch effects. So what we observed is that as soon as we increase the number of prior components from 1 to more than 1, the both biological preservation, but also the batch correction improve. So we wanted to understand why is batch correction better, because this was something unexpected. So we look also at its sibling, the Gaussian mixture prior. And what you can see here also, when you increase the number of prior components, in generally the integration improves, but never so much as in the Venn prior. And also, if you can see, if you increase the number of prior components too much, the performance drops again. Therefore, the Gaussian mixture prior is less robust. So what we wanted to look at then is also how do prior components in the Venn prior correspond to individual cells. What you can see here, we assigned every cell to the prior component with the highest probability. And we observed that every prior component attracts cells from only a subset of cell types, but from both batches, which means that the cell types can remain separated while the batches are mixed. Therefore, the Venn prior enables better batch correction while preserving biological variation. Another approach that can be used for improving cell type preservation is cell type supervision. So what happens here, we use prior knowledge about cell type labels to improve cell type representation. This is an interesting approach. However, it has a couple of limitations. First of all, we actually need to have prior labels known beforehand, which is not always the case because this requires a lot of manual work. Second of all, even when we have them, if we're comparing different data sets, the labels might not be matched across data sets. So we again need to invest some time to match them. And even so, the labels might not be completely truthful, so they might lead to biases in our integration. Therefore, we didn't really focus on this approach in our paper, but I'm nevertheless going to show you a couple of examples because I think it's interesting. So if you're interested, feel free to explore it in the future. So there are two key strategies that can be used. One of them is contrastive learning, and the other one is classification-based. In contrastive learning, what we have cell types, and within every cell type, we would try to push the cells together, and between cell types, we try to push the cells apart. In the classification, we basically just have a latent space, and we want to make sure that we are able to classify the cells based on their cell type of origin. Now, one issue that occurs in contrastive learning is that the cells within a cell type are pushed together, which means that we can actually destroy within cell type information, especially if our biological variation is present at a cell type resolution that is finer than what we have available for supervision. Basically, everything is pushed together, although it might not necessarily be biologically meaningful. On the other hand, in classification, we only want to make sure that the cell types separate nicely. Therefore, we're not disturbing the within cell type structure in any way. This is just one of our observations, and of course needs additional validation, but I'm going to show you one example how we often see that, although further metrics and analysis would be needed. So here we have one integration that is done with just unsupervised cVAE model. We have one that is based on classification model and another one that is based on contrastive model. What you can see here is that in the unsupervised one, a lot of the cell types kind of mix together or are located very close by, making the downstream analysis harder. And then when we look at the classification model, those cell types separate much more nicely. On the other hand, we can also see that there's some finer within cell type structure within some of the cell types in both models here. However, when we look at the contrasting models, this finer structure is lost. Therefore, while supervision can be beneficial, we must be also careful that we use an appropriate approach for that. And I would here say that our observation needs further validation. So if you're interested, to feel free to explore it more. Now, to summarize, what are the important things about biological preservation? One prior is here the approach that we chose because it enables both good biological preservation and batch correction, and the cell type supervision can be helpful, but requires more further work. So now let's look at how we can combine different approaches to further improve our integration. So here we are combining two complementary approaches, the cycle consistency, which is going to push together batches, and the Van prior, which provide us a more expressive latent space. So what we wanted to see is if this combined approach actually improves upon the base models. And for that, we looked at the trade-off between batch correction and biological preservation, which we want to optimize. So what we do, we increase the batch correction strength by increasing the parameter that regulates this batch correction. So that would be the regularization objective in the Van prior and the cycle loss in the cycle model. And then we looked at the correlation between the batch correction hyperparameters and the individual batch correction and biological preservation metrics. So what we want to see is as we increase batch correction hyperparameters, that the batch correction increases, so has positive correlation, but the biological preservation does not drop, so therefore having a neutral correlation or not having a degradation. What you can see here is that Van prior has the highest positive correlation with batch correction and also the least negative correlation with biological preservation. Therefore, we believe that combining those two approaches actually enables us to achieve a better trade-off between batch correction and biological preservation. Now, one very important thing we need to do at the end is also to make sure that our integration is meaningful. And one of the things we want to do downstream is probably compare different samples, because that is what we did batch correction in the first place, to be able to compare different samples on a shared embedding. So here I'll be showing you one example of data set from retinal organoids and 2 regions of retina tissue. The authors from the study reported that the organoid tissue is quite similar to the tissue periphery. However, those 2 populations are quite distinct from the tissue fovea, which is also marked by expression of a specific marker. Now, this directly corresponds to what we observe in our VAMP and cycle consistency model. However, if you, for example, look at the adversarial glue model, the organoid data is mixed with both regions, also the fovea one, which is biologically incorrect. So if we want to do reliable downstream analysis, we need to have high quality integration at the first place. Now, just to summarize what you need to know to improve your integration. So vampire and cycle consistency offer good batch correction and biological preservation trade-off. They can be easily added to any other cVAE model. So if you're working on something related, I definitely urge you to try it out. And if you're not into developing your own model, we also made now the model available as cVAE in the scVAE tools package, which is commonly used for single cell integration. Of course, we did not answer all the questions that are about integration, so there's quite a lot of things, but those 3 are most interesting to me. So here I worked with data sets that have substantial batch effects, so cross-species, different technologies, organoids, primary tissue. However, the question is if the same approaches, so the vampire and cycle consistency, would be also beneficial if you compare data sets with weaker batch effects, for example, just patient populations generated from different hospitals. The second question is, can we add any other approaches that would further improve our model? So as I already hinted, supervision could be useful. Another thing that might be especially interesting for cross-species integration is looking at flexible feature relationships. So in different species, we have different orthologs that don't always map to one-to-one. So we might want, instead of one-to-one feature mapping, having some kind of graph to represent the relationships. And also the orthology information is usually based on protein sequence, which does not directly correspond to expression. Therefore, having more flexible relationships between features during training could be beneficial. And there are already some models out there that try to explore those approaches and use different strategies to do that. However, again, I think there would be some additional work needed to explore which of those strategies is actually the most beneficial and in what kind of data settings this has any benefit at all. Now, lastly, I here focus on single-cell transcriptomics. However, it is possible that these models could be also applied to other modalities. So for example, there was already work testing of whether single-cell integration models could be applied to image features. And if you have any data sets where you think that this model might be applicable, I would be definitely interested from hearing about you, whether it worked or not, and what are the things that might translate to other modalities and whatnot. Now, at last, I would like to thank my collaborators, specifically Amir, who helped with matchmarking and analysis of the vampire, and Luke, who provided me mentoring, as well as everyone else from Thais and Kelly's lab who collaborated on this project, and everyone who funded my work. Now, thank you for listening, and I hope that this talk will help you in your own data integration. And if you have any questions, I would be happy to answer them.
Cool. Thank you for the talk. No questions right now in the chat, so I guess I can start. In the vampire, how do you choose the I forget how they're called, but like the representative samples which define the modes?
Yeah. I guess I don't need to skip, but basically what we observed is that this can be really done at random. So one thing that is important is that you initialize it with true cells, because if you initialize it with just random parameters, that will be too far from your data distribution and therefore will lead to challenges when you do the training. However, if you just pick any of the cells, that will work well. We tried initializing it with like one cell type with balanced way, across cell types with different batches, and it didn't have any effect at all. So we figured out it is really robust, and also the number of prior components, as I showed, was quite robust. So ranging from like, I don't know, a bit more than 2, that is still kind of on the border, maybe like 5 or so, to also larger numbers would be fine. But because you don't need larger number, because fine is completely fine, you don't need to fit more parameters. So it seemed quite robust.
Cool. And for the, I guess, contrastive type losses that you've tried, are there like, well, I guess I have 2 questions. Like, what's the proportion of data that has good labels? And then are there only these cell type labels, or do you have more labels that could be interesting?
So here, we usually, often when you use datasets, you have most of the cell types annotated. Of course, those approaches don't need to have all the cell types annotated, and we didn't do any screens that would comprehensively look at, what's the proportion of cells. So this is that is needed to improve the representation. So this is something for the future work. And then you mentioned if there's any other information that could be used. So I know there are out some models that try to learn cell type information in a hierarchical manner. So for example, you have immune cell, but within immune cells, you have lymphocytes, and you have eosinophils and so forth. And then you can kind of, guide the model also to place together cells that should be more similar. That's the only approach I have heard so far, but it's possible that you could add any additional information, such as, biological information on coming from different conditions. But then again, you come into the danger of actually enforcing information on whether those cells actually separate or not. So if you say my model needs to be able to get a latent representation when healthy and disease separate, you come into the danger that you're putting things apart that might not be separating. So maybe the disease is affecting only some of the cell types and not all of them. So you need to be a bit careful on what kind of priors you choose there.
Yeah, that makes sense. Ali?
Hi, great talk. Thank you so much. We'll totally be trying this out. Can I ask you to tell me a little more about the data sets and how many data sets you were testing this on? I mean, you said it was things with substantial batch effects across species, but yeah, how many, how far apart those species? And if you did any like purposeful engineering of this, like you took real data and then removed complete cell type or cluster from it to see how this affected it.
Yeah, so we started from completely natural data sets. We didn't do any engineering. And then we look at what actually happens with those data sets when you use different integration methods. So the project kind of started like a benchmark. And then when we needed to wrap it for the paper, it was more like a method. But basically, yeah, we tried to observe, what kind of things go wrong. So what I tried on, I tried to get 3 representative examples. So I had cross-species mouse and human pancreatic data sets, so pancreatic islets. I had adipose tissue where we had single cell and single nuclei. This data was actually from one study, but still often people have challenges to integrate those. And then we had another data set that was the retinal organoids and the primary tissue. We tried this model on a couple more data sets, but we didn't run any benchmarks. So it was more like for internal use if someone wanted to get an integrated embedding. So we tried on some additional pancreatic data just because me and my collaborators work a lot of pancreas data, so with organoids and embryonic development. Then we were looking with another brain organoid data set. And I know that my colleagues are now trying it on some other data sets, but those results are not finished yet. So I don't know how exactly it went there.
Great, thanks. If I can squeeze in a follow-up question. Sure. One thing that caught my mind that people want to do with this, so I'm just curious if you're aware anyone's trying it, is, I mean, you could say like proteomics versus transcriptomics is a substantial batch effect, probably a little more than single cell versus single nucleotide, but have you heard of anyone trying to integrate those two?
I think someone in our lab was working on prediction across those. So we could probably get the data to try to integrate it. I know there are specific methods who try to do cross-modality integration. I think in this specific case, it would be actually fine to just map features one-to-one because you work with proteins. But if you were, for example, to work with another modality such as, ATAC-seq, where it is not so straightforward to map the peaks to genes, then maybe using a custom model that models both of the modalities separately, so having different features could be more beneficial. But I definitely think that it would be worth trying it out if you have expression and proteins, especially because our model here works with normalized data. So it doesn't assume that there would be any specific data distribution. So for example, some of the integration models, they will assume negative binomial distribution working with raw single cell data, which might be not appropriate for protein data, but we focused here, we just assuming that data is normally distributed approximately, which usually happens for single cell data after you normalize it and log transform it.